Geneticists would like to understand genes. Now, those genes have to be in an organism, at least originally, or a virus. And so, therefore, some geneticists study bacteria, just as some geneticists study fruit flies or yeast or mice, etc. There are lots of reasons to study prokaryotic cells, bacteria. One is the incredible number of them. They are the most abundant organisms on the planet. Even if we were to consider the human body, we carry about 2 to 3 pounds of bacteria around with us. But because they're smaller cells, the number of the bacterial cells associated with our body actually outnumbers the number of human cells. And so when you look in the mirror, there's actually more bacterial cells in that mass than there are human cells. So clearly, bacteria are important from a numbers standpoint. Also, just from a biomass standpoint, just a weight standpoint, while plants represent the majority of life on Earth as far as weight goes, if you were to weigh the amount of life on Earth and say, here are all of the animals, notice that all animals, including all humans, are outweighed by protists, but that all protists are then outweighed by a group of bacteria known as the archaea, which we often find in more extreme environments, but they can even be found in our intestines. So notice how many of them that there are. But then when you consider then the other bacteria, the eubacteria, the more common group, that they far outnumber the other groups. And so this, while no bacteria equal the number of plants representing 450 gigatons, there are 70 gigatons of bacteria and 7 gigatons of archaea. So that's the line for bacteria, that's the line for archaea, far outweighing all animal life. And so there's a lot of reason to study genomes of bacteria. They're the most common living things in terms of number. Other than plants, they are among the most common when it refers to just the entire mass. And there's so much about them we don't understand. So for example, we don't know how much of them live in the extreme environments of the Earth and the deep biosphere. And we often think of life as being odd if it lives near 100 degrees Celsius, or if it lives in rock deep under the Earth or deep under the Earth's crust. But because there is so much bacteria there, now they're living very slowly, their metabolisms are very low, but nevertheless, does this represent half of the bacteria on Earth and a third of the life on Earth in these extreme environments? We just don't know. And so geneticists, there's a lot of reasons to study bacteria in general. But I would like to focus a bit more on studying them just to understand genes. So even if you're not that interested in understanding bacteria in general, nevertheless, we can understand genes, including human genes. As I'll discuss later, bacteria, they were the first form of life on Earth, so that they first appear 1.5 billion years before anything that's not a bacterium. 3.5 billion years ago, there were bacteria. Eukaryotic cells don't appear until about 2 billion years. So for a third of the history of life, then these organisms had the planet to themselves. And when we want to study, big complex organisms like ourselves, we find that many of our enzymes, like the serine proteases, which digest protein, they started in bacteria. The globins, which we use to transport oxygen, started in bacteria. So many of our genes date back to that. So very often it's by identifying genes, getting the sequences of genes in bacteria, that we can then look at human cells and say, do humans have a version of this gene? So even if you're interested in studying human genes, very often you start off with bacteria, finding it there first. Now, bacteria are what are called prokaryotic cells. And in general, I'm sure every textbook you've ever referred to, will say that these are smaller and simpler. They don't have a membrane-bound nucleus. They have a circular chromosome. Now, what I just said, that's not wrong. All right, so it's not wrong that when thinking of, say, eukaryotic cells like animal and plant cells and comparing them to bacteria, to say that bacteria are smaller, they're simpler, a membrane-bound. I'm sorry, they lack a membrane-bound nucleus. They have a single circular chromosome. But the reality is that's just a generalization. Bacteria are incredibly diverse, and therefore some of them are larger than many eukaryotes. There are bacteria that you can see without a microscope. When it comes to the number of genes and how big their genome is, there is an incredible variation. We have 13 megabase pairs, 4.6 megabase pairs. The number of genes varies greatly, as you will see. Also, how many chromosomes do we have? Some bacteria have a single circular chromosome. Some have rod-shaped chromosomes. Some have circular and rod-shaped chromosomes. Some, in addition to that, have plasmids. So here you can see, just in terms of the number of genes, while the others could have thousands of genes, here we have under 1,000 genes, a much smaller amount of DNA. I'll get to the big ones in a second. And so bacteria aren't all the same. And so any generalization that you say, this is their size, et cetera, getting to their chromosomes. Once again, they can have a single chromosome, they can have multiple chromosomes, they can have both circular chromosomes and linear, the way that our chromosomes are, the rod-shaped ones. There can be plasmids in addition to the chromosomes. These are loops of extra chromosomal DNA. Some even wrap their chromosomes in a membrane. So we often say that the eukaryotes have a nucleus while the prokaryotes do not. And while it's true that the double membrane, which we have in the eukaryotes, we don't see that there. Nevertheless, you can see a membrane-bound nucleus, or I'm sorry, a membrane-bound region where the DNA is in some bacteria. Some of the largest ones, once again, you can see this one is 3/4 of a millimeter in length, so you could see it without a microscope. It's polyploid with thousands of copies of its chromosome. So that being said, we often, in a general sense, try to portray the genome of prokaryotes in this simple way. But prokaryotes vary. Once again, they had 1.5 billion years to diversify before there were any organisms that weren't bacteria on the planet. So there's certainly a great diversity there. One final point, we often study the microbes in lab, which can grow in labs, right? So, anything that requires extreme heat or extreme acid levels or salt levels, et cetera, it's just harder to keep those. So we tend not to study those. But it should be just a reminder when we study genomes and genetics, if we're just studying those things which live in lab conditions, you're omitting much of the deep biosphere, which once again could be 1/3 of life on Earth, including, if we were to compare sequences, what might have been the earliest organisms on the planet. And so eukaryotes, they are all related to each other, and they first appeared on the fossil record 2 billion years ago. The two groups of bacteria, the archaea and the eubacteria, they go back earlier than that. And the earliest branches of both the archaea and the eubacteria, these are the ones which live in extreme environments today. So, very hot temperatures, great pressures, etc. And so it would seem that the early Earth was different than it is today, and that these extreme environments with these extreme forms of genetics, this is actually, what the first living things were. So once again, we should not study those. And then even in biotechnology today, so for example, with PCR, you need an enzyme which works at boiling temperatures. And the Nobel Prize was won by the individual who said, oh, let's just take the DNA polymerase from these bacteria which live at very high temperatures, and then we can use that. And so there's so many reasons for geneticists to study bacteria. Now, whether it be studying bacteria, studying disease, or biotechnology, a couple of important applications are how do genes move from one bacterium to another. So bacteria do not undergo sexual reproduction. So no bacteria have true sex where haploid cells fuse to make a diploid cell that undergoes meiosis to produce haploid cells again. But there are three ways in which genes can be exchanged from one bacterium to another. And these are then important for the study of genes and also biotechnology as new organisms with new traits are engineered. The first is what's called transformation. Now, bacteria can take up DNA from their environment. So let's say here's a bacterium and it dies. That could be actually any cell. That could even be a human cell that dies. And so here's the DNA from the dead cell. A lot of bacteria will ingest this using the DNA from a previously dead cell as both an energy source and a source of carbon. So a lot of bacteria feed on the remains of the biomolecules from dead organisms, so decomposers in that sense. Sometimes, however, if they take up foreign DNA, instead of using it as food, if it seems to match a section that they have, they can actually then keep this DNA and then undergo recombination, cutting out their own gene and inserting this foreign gene in its place. So now some of this bacterium's chromosome was what was ingested from the environment. So while they don't have sex, this organism has just got DNA from the dead. And so a dead cell has contributed a gene which might give a trait to now this living cell. And whenever this cell divides now, it will replicate that foreign DNA along with it. So this happens all the time. And this is, from a health standpoint, why you don't want to broadly and unnecessarily prescribe antibiotics, because if you have dead antibiotic-resistant bacteria in your large intestine, you might say, oh, but that's okay. That's one of those normal bacteria. I don't mind they're antibiotic resistant. But yes, if they die, here's a bacteria that might cause a disease. It could take up that DNA, and now this bacterium could be antibiotic resistant. Many bacteria can take up DNA even from other species. They're less picky about where they're getting a genetic exchange from. And so now bacteria could have new traits. So this is important from a health standpoint, but also from a point of genetic engineering, where you could say, I would like a bacteria with this trait. Maybe it could make something that my industry needs. Maybe it could make something the hospital needs. I'll give it a human gene, and then it can then take up that foreign DNA and get a new trait. So this is one way that bacteria can get new DNA and thus new traits, through what's called transformation. DNA from dead bacteria can be taken up and incorporated. Now, geneticists, when they try to find genes, often then are interested in mapping. So how do you find genes and the order? Well, you could actually use transformation in this process by asking, all right, let's say there's two genes that we're looking at. What is the rate at which cells will take them both up at the same time? What is the rate at which cells will undergo co-transformation? And the idea is that if two genes are very close to each other, the odds that a cell will take up this foreign DNA both together, then it increases. If if they're farther apart, that is less likely. And so if you're trying to map genes, you could then use the frequency of co-transformation, how frequently genes are undergo transformation together, with the idea, once again, that if they're close to each other, then they're more likely to undergo transformation together. If they're farther apart, they're less likely. And so if you're interested in mapping, then you can use transformation. Before I go on, so here's just kind of a simulation of, say, how a transformation might occur. So let's say you have bacteria. Now, once again, this can be done in a lab, but this could be going on in your intestine right now. Bacteria can take up foreign DNA. So if we have a bacterial broth and we have bacteria with a certain trait. So bacteria have traits, some can grow on this medium and some can't. So I'll mention that again. A bacterium which needs certain things in order to survive is called an auxotroph. So you have to give it certain nutrients or else that bacteria can't grow. While in contrast, a bacterium which has all of the things it needs to grow can be called a prototroph. Also, some bacteria can have a trait whether they're antibiotic resistant or not, or have enzymes that do certain reactions. So let's say that here we have a broth with bacteria which are missing two traits. There's two things that they can't do. But then we have DNA from dead cells with two genes, all right? And so we have DNA, and we're going to add this to a plus tube. So the broths are the same, but the plus tube is going to get DNA from a dead organism that the minus tube does not get. So for example, these cells, they could be missing a gene for ampicillin resistance. They could be AMP-R minus. They could be missing a gene to turn lactose into a blue product. And so an enzyme could then perform a chemical reaction, which then would show a visible result. You could actually then see, this bacteria makes this enzyme because when I give it this galactose analog, the gene that it's making, beta-galactosidase, makes a blue product, all right? So that there are now two things we can look for in these bacteria. We can look for whether they're resistant to ampicillin or if they can make a blue product. The minus tube did not get DNA with these genes. The plus tube did. Now, in our procedure, we might perform a few things which make it more likely that the cells will undergo transformation. Transformation turns out is a rare event, and we want to encourage it. So we could give them calcium chloride in the solution that they're in, and we could give them a heat shock where we take them from a cold temperature to a hot temperature for 90 seconds, and then back to a cold temperature. So this is what's called a heat shock. And these things make transformation, which is a rare event, it makes it more likely to occur. Gets the bacteria in the mood, in other words, to undergo a transformation. Okay, so If the bacteria have undergone these things which make transformation more likely, then transformation should only be occurring in the plus tube. The plus tube had that foreign DNA. The negative tube did not. So in the plus tube, these cells could be taking up this foreign DNA. The minus tubes aren't. And as a result, there might be some cells which then have new traits. They might then have the ability to live in ampicillin, which the normal cells do not. They might have the ability to make blue product, which the normal cells do not. And so if we were to do this experiment, we would expect that both the plus and the minus strain could grow on plain Loria broth agar. Loria broth would just be food. And so we would expect that the bacteria could grow just fine here, both the plus and the minus strain. But if we add ampicillin to the agar, we would expect that all of the minus strain would die because none of them got the gene for ampicillin resistance. And if we have a plate with ampicillin and a galactose analog that could be turned blue, once again, we expect the minus strain to die because there's ampicillin here. So we expect no growth on the minus strain here. But if the plus strain has undergone transformation, we expect cells to be able to live in the presence of ampicillin, and then on the LB-AMP-X gal plate, not only for them to be able to live, but to be able to make some of that blue product. Now, how rare event is transformation? Well, that depends. I mean, if it was common, you'd see a lot of these. If it was rare, you would see fewer. So that's something that we could measure and actually then count. We could even then ask about co-transformation. If there were two genes living with ampicillin, so the gene for resistance, plus the ability to make that blue product with the enzyme beta-galactosidase. Here is a cell which got one of the genes, but not both. It got the gene for ampicillin resistance, but not the gene to make a new, that blue product. This then is a sign that these genes don't always undergo co-transformation together. And once again, the frequency of that could be used in mapping. Now, if a bacterium got one gene for beta-galactosidase, but not the gene for ampicillin resistance, it'll die. And so this isn't measuring all of the colonies which have one gene but not the other, only the ones which have the ampicillin resistant gene and not the other. As I go through in this video, if you were doing this experiment and you were concerned about error and you wanted to run some controls, there would be unexpected results, which would indicate, contamination. So when you're working with bacteria and genes, things that you can't see, you assume that what is in your vial is what you expect it to be, but you always need to do controls just in case. So that's transformation. And like I said, sometimes after transformation, these bacteria are worth an incredible amount of money because you gave them the gene to, say, make human insulin, which then you can send, to hospitals for, diabetics, et cetera, as opposed to the old days where you'd have to go to a slaughterhouse and get animal insulin. And so I'll address that later with genetically modified organisms. But you could, say, engineer bacteria. If you want bacteria to get rid of sulfur from acid rain or acid mine drainage, or bacteria which can help eat an oil slick, and clean the environment, you could engineer bacteria with new enzymes that can do this, that could, hopefully someday make petroleum. Right? I mean, petroleum's a molecule. I mean, if bacteria could make it, we wouldn't have to dig for it. We could just have, say, blue-green algae make it in sunlight. And so if you could engineer the right bacteria, they would be worth a lot of money. A second process through which bacteria can take up new genes is what is called transduction. Normally, viruses inject their nucleic acid, they destroy the host genome, and then turn the cell into a factory that makes, say, all of this viral nucleic acid, which I will depict here in white. This white viral nucleic acid, this could then be packaged into a viral particle and thus infect another cell. However, sometimes some of the host chromosome is accidentally packaged in the viral package instead. All right, so the virus intended for white viral nucleic acid to go inside this virus, but by accident, a yellow bacterial strain was put, strand was put in there instead. And so now as a result, the next time that this virion infects a cell, it's not going to inject viral DNA, but rather DNA from the dead cell. So a new living cell will get new genetic information from a dead cell, and it might end up getting a new trait as a result. This is what's called transduction. It's also called generalized transduction because it was just dumb luck. Any part of the host chromosome could have been accidentally packaged in the viral particle. There's also what is known as specialized transduction, where some genes are much more likely to undergo transduction than others. And the reason for this is some viruses insert themselves into the host genome for a while as a temperate virus, and then can become active and become lytic again. But they could then have, say, red genes, which were the host genes next to the viral insertion site. So the virus just didn't insert to any part of the host chromosome. It inserted into a specific spot here where there were red host genes. And so the yellow genes are coming from anywhere at random. But if the virus inserted to part of the host chromosome here, then if red genes end up being in the viral particle, because when this decides to go from the temperate life cycle to the lytic life cycle, it sloppily cuts itself out. So this is what is then called specialized transduction, where the virus is going to transport host genes, but not at random. It's transporting the host genes which were next to the viral insertion site. The third and final way that you can have a genetic exchange between two living among bacteria is by having an actual exchange between living cells. This is what's known as conjugation. So in transformation, bacteria can take up DNA from dead cells. In transduction, the virus helps to transport host DNA from one cell to another. But here, in what's called conjugation, you can have a genetic exchange between two living cells. Often what's involved is what is called a plasmid. Okay, so plasmids are extra chromosomal loops of DNA. So as I had mentioned earlier, some bacteria just have a chromosome. But you can have other versions of bacterial DNA, including what are called plasmids. These are extra chromosomal loops, DNA that's not part of the main chromosome. And sometimes you can have multiple ones of these. Well, one which is well studied is what's called the F plasmid. The F plasmid will then code for the genes necessary for a cell that has the F plasmid, let's call that F+, to then make what's called a pilus. Lots of bacteria, they have extensions of themselves which allow them to adhere to the environment, sense the environment, called pili. So a pilus could be made which causes the F+ cell to attach to an F- cell. The plasmid then replicates itself, and one of the two copies then passes through a pore from the F+ cell into the F minus cell, but now the F minus cell has an F plasmid. It is now F plus. And so this plasmid in this state, just an F plasmid, not the F prime I'll talk about in a second, it doesn't add anything to the phenotype of the bacteria. It doesn't help the bacteria live. It's not, giving it a new trait. It instead is simply coding for its own propagation. It is selfish DNA. It's not helping its host organism. It is simply propagating itself because it can now make F minus cells into F plus cells as it passes from one cell to another. Okay. So that is what's called conjugation, because it is an exchange between two living cells, and that involves the F plasmid. But then there are variations on this. So for example, some F plasmids now have an additional gene involved, say that green gene. And so thus, if this is a gene for, say, ampicillin resistance, or a gene to, make a specific enzyme, now when the plasmid is transferred from, the F plus cell to the F minus cell, it's not just bringing the DNA for a new plasmid, but rather it's also bringing a gene which will give a new trait to this cell. So maybe this cell now can live in an environment where it couldn't before, because now it doesn't need a certain nutrient. It can make its own. Maybe it can now live in the presence of an antibiotic or whatever. So an F prime plasmid has another gene which will now add to the phenotype of the host. One third type of I'm sorry, before I get to the HFR plasmids. And so one can then look for conjugation. So let's say that now we have two strains of bacteria. Let's say that strain 1 can survive in antibiotic 1. Let's say strain 2 can survive in antibiotic 2, but that neither strain can live when both antibiotics are present. And so if we were to set up an experiment this way, that the plain Loria broth, once again, this is just agar with food in it, that both strain 1 and strain 2, strain 1 is, say, on the left, strain 2 on the right, that both of them can survive in this environment because it's just plain food. Let's say that when we have our first antibiotic, ampicillin, strain Y can survive that, but strain X can't. Let's say when we add another antibiotic, streptomycin, strain X can survive that strain, but strain Y can't. So neither of them would be able to then survive an agar plate that has both types of antibiotics. So if you have a plate with both ampicillin and streptomycin, then no growth should occur. But if they undergo conjugation, if one of the genes is on a plasmid and can be transferred from a one living cell to another, you might then, after, on a mating plate, all right, where you have strain X and strain Y, but then you mix them, you take your loop and then take some of the cells from here to there. Now you have direct contact between living cells, and you might have a plasmid transferred from one strain to the other, which then would mean that now you have some bacteria which not only have a gene for the resistance to one antibiotic on their bacterial chromosome, but now a plasmid for antibiotic resistance on a plasmid. And so now you should be able to find cells which can live in both. So the cells over here died, the cells over here died, the cells up here died, but where they contacted each other, you had conjugation where plasmid was transferred, and so now you have bacteria with a new trait. All right, so that was just an experimental demonstration of conjugation. A third kind of plasmid is what's called an HFR plasmid, a high frequency of recombination. In this DNA, the DNA is inserted then into the host chromosome. So recombination can say, if there's this sequence in the host chromosome and this sequence on foreign DNA which has come in, recombination could say, oh, but they're similar, and so the host DNA is cut and this is inserted instead. So what started off as a plasmid could then actually be inserted in the host chromosome, but at a specific spot. And so if you're trying to map genes, and as I had said elsewhere, if you know where a gene is, you can get its sequence. You can then say, oh, do humans have a version of the gene? What kind of protein does this encode? Once you know where a gene is, there are lots of genetic techniques that you can now use. So there's a lot that you can do. If you don't know where the gene is, then now you're very limited. There's much less that you can do. And so one of the things that you want is to find genes, to map genes, to say, where is the gene that does this? And so let's say we know that bacteria have a purple gene that makes this enzyme, a blue gene involved in DNA mutation repair, an aqua gene, a green gene. We just don't know where they are. And until we know where they are, we're limited in what we can learn from these genes. But here's a way of mapping the bacterial chromosome so that we know where the genes are. So in an HFR plasmid, the plasmid inserts itself into the chromosome. And if conjugation occurs, what happens is that the plasmid will start to transfer itself. But because it's attached to the host chromosome, it actually cuts in the middle and then starts to drag the host chromosome with it. And so if you gave it enough time, the whole host chromosome would end up in the second cell. And then you could get potentially recombination, where the blue gene or the purple gene from cell number 1 got incorporated into the chromosome here. But let's say that you don't let this whole process play out. If you gave it enough time, all right, maybe a couple of hours, then the whole chromosome could be transferred, and you can see recombination. I'll say how you could demonstrate that in a second. So while that could happen, what you could do is what's called an interrupted mating experiment. The original ones were done with a blender, so that you could let these cells start to undergo conjugation, all right, but then just hit start on the blender, say after, say, 10 minutes, all right? So after 10 minutes, if you hit blend, then conjugation started, but it couldn't get very far. And so you might notice that you did get one gene transferred from the first cell to the second, but it was just the purple gene. But that's good. You now know that the purple gene is the one that was right next to the insertion site. If you got the purple gene commonly and maybe a few cases of the blue gene, then you would say, oh, that's the next one as we go on the insertion site. So we're going to start drawing a map of the bacterial chromosome, starting with the origin, the site where the plasmid has inserted itself. And then we will now start saying, oh, this purple gene transfers first. The blue gene transfers second. Now, maybe now we'll give it 20 minutes before we hit the blender. And then you'll see, oh, the next gene is the aqua gene. And then we'll give it 30 minutes and then 40 minutes. And then after a while, then you've mapped all of the genes on the bacterial chromosome, which is now good because you can get the sequences, and you'd say, oh, you know, the gene that's fixing DNA, it has this sequence. We can now look for it in humans. We can now see if it's traveling through a human family as a source of cancer. So there's so much you can do once you've found it. And so what we're doing here is we are looking for which genes are getting transferred after 10 minutes, after 20 minutes, after 30 minutes. The ones which are closest to the insertion site get transferred first. The ones which are farther, they need more time. And this is where some of those bacterial phenotypes come in handy, because obviously if you want to see if this cell, the second cell, got the genes of the first cell had to have a different form of the gene than the second cell, so that you could say, oh, like, in humans, you could say, oh, look, an individual that had brown hair now has red hair, or an individual that had blood type A now has blood type O. You could see, a trait was transferred. Well, what could we do with bacteria? Well, bacteria, you could transfer something which would change the shape of a colony. You could transfer something for antibiotic resistance. But very commonly used ones are for nutrient requirements. Let's say that the first bacteria, the one on the left, that was a prototroph, which can make all of its own nutrients. It can make its own histidine. It can make its own methionine. It can make its own lactose or whatever, so it can, everything that it needs, it can make. So we would call it His plus, Met plus. It can make its own histidine. It can make its own methionine. It has the genes for those enzymes. In contrast, this second cell, let's say that it was Met minus, His minus. It has to grow on an Auger plate that you have added methionine to, or histidine to, or else it can't grow. So it's what's known as an auxotrophic strain. It needs stuff in order to grow. All right, like humans, we can't make our own vitamin C. We have to ingest our own vitamin C because we can't make it. So this bacteria might not be able to make a certain nutrient. But after undergoing conjugation, now it can. So you could say, all right, so this bacterial strain couldn't make its own methionine and needed for it to be in its environment. But now, after conjugation, it now has a new gene and it can make its own methionine or histidine or whatever. So that would be a way of measuring the transfer of these genes, because bacteria, they do have phenotype. And so thus when it comes to mapping genes, bacteria are useful. Now you might say, oh, well, can't we just use fruit flies and map them there? Well, maybe, like for one with, that affects fruit fly eye color. But how about one that affects DNA repair, right? Because if the fruit flies can't repair their DNA, well, they might just be dead. So you actually, for some of these essential enzymatic pathways and essential reactions and processes involving DNA, that you can't use mice or fruit flies, that you have to use simpler organisms like bacteria. So some geneticists, they study bacteria. Even if they want to study humans, all right, they find genes in bacteria first and then take what they know and apply it to humans. We know an awful lot more about human cells, human metabolic reactions, human genetics from the study of bacteria. Sometimes you want to engineer a bacterium to make money. So let's say here is a bacterial piece of DNA. We apply a restriction enzyme, which I'll talk about later, and we cut it so that we have, a specific overlapping sequences, these sticky ends. But now if we apply the same enzyme and cut human DNA, now we have human DNA, which has the same sticky ends as the cut bacterial chromosome. You could actually then insert the human DNA into the bacterial chromosome. Or maybe you could put it on a plasmid that could be used then for conjugation or transformation. And now you have a bacterium which makes something that humans want. Maybe there's something that you want to, give to hospital patients, like interferons or whatever. Well, you could get it from human urine. You could try to isolate it from, human blood. You could try to get growth hormone from the pituitary glands of human cadavers. You could get insulin from other animals from a slaughterhouse. Or you could just engineer bacteria to make a human enzyme, or an enzyme that could then help you with the factory that you're running, generate a product that is desired. So a lot of bacteria are being genetically modified to help with industry or to treat patients, etc. Now, I'm going to get into it a little bit later. It's a little beyond this first introductory topic, but there's a technique known as CRISPR. Actually, it comes from bacteria, where it was originally a way that bacteria could remember what viruses have infected them in the past. Now that's pretty impressive, because humans, we make a big deal about our adaptive immunity, which lets us learn as we go. But bacteria can actually have a version of this as well, where they can get infected by a virus and then learn from that so that they will be less vulnerable to that virus in the future. And in doing so, they can actually change their DNA. Well, we humans can now use CRISPR to change DNA and then to now generate organisms with a new trait. So in this case, maybe bacteria that can do something that we'd like. So for example, if we could generate bacteria which could, say, make petroleum, we could solve climate change. We could get petroleum in a renewable way, not by digging up old petroleum, which would now add more heat absorbing carbon dioxide. We could just absorb carbon dioxide from the air and make petroleum from that and return it, and then make it as part of the carbon cycle. So that would, potentially have planet altering consequences if we could engineer the right bacteria. So there are lots of reasons for geneticists to be interested in bacteria, whether it's just understanding life on Earth, whether it's as a way of better understanding humans and our DNA, because a lot of these genes first evolved in bacteria, or maybe we can map them first in bacteria, or if we're interested in now generating new organisms which can do new things, like make us molecules which we humans want, or help clean up our pollution. If we can get bacteria which can sequester heavy metals or break down oil spills, etc. So once again, a lot of geneticists study bacteria, and this lecture was just kind of an introduction into some of the principles of that.